Hi, I hope you're doing good. So this video is just a short kind of like overall description about our protocol in go and get cloning. And so as part of molecular biology, cloning is a process where we make identical copies of like genetic material. And in our case, we're trying to make many copies of our CRISPR genetic machinery. Okay, and we're using go and get cloning. Why go and get cloning? Go and get cloning is cool because it actually allows us to do many different like two major processes in one tube, one PCR tube. And so in our case, it's where like we have like this original CRISPR genetic machinery, which has all the components to like allow the CRISPR process to take place. Where like the guide RNA once this piece is introduced into an organism, the guide RNA components are going to kind of like search the genome for like the target gene or the complementary sequences. Once they reach where like that target is, then the Cas9 protein, which acts as a molecular scissors, is now going to cause double-stranded break in that genome area, and then that part will lose its function because the repair system is going to make errors. So this original plasmid, it's actually intact. It's like it's undigested. So we remember that we actually did mini prep. When we did that mini prep, we're able to extract this original plasmid. So that undigested plasmid, we need that, but then we need to add our own CRISPR components, our own target, and so that are the ones that we did with PCR, then we PCR purified. So this this original intact plasmid has to be digested, and that's why we have like our enzymes. We're going to cut, okay, at particular places, because the association enzymes cut at particular places. They recognize certain sequences, so it's going to cut, okay, our intact plasmid. And then so that's like the first big process where like the cutting digestion is happening in this tube. And then our pieces, our purified PCR pieces, which have like our specific sequences, target sequences, will then join. And when they join, they have to be kind of like sealed together or ligated, okay, put together. And so the next big process is now like ligation. And so both digestion, the cutting, and the addition of the components happen in one tube, which is really cool because back in the day, molecular biologists used to do these things in separate tubes. They have a separate tube for just the digestion part and a separate reaction or tube for the ligation part. And so for us in our case, if we look at the protocol, T4 ligase enzyme is what does like the ligation. Restriction enzyme does like the digestion. Okay, and so how to do this this go and get cloning reaction is that we just have to have all these components together. So we get an ice bucket, we get all these different reagents from the freezer, -20. We have two enzymes in our case, the T4 ligase. We also have to have ATP because this actually needs like energy, and also a buffer. The buffer is like to keep like the conditions perfect. Because remember that this is a process which happens naturally in the bacterial cells. Bacterial cells, in the cells, they know they like at some point they have to cut the digestion of like certain DNA happens naturally, and so in the cell there's a particular environment that needs to be maintained. We're doing this in vitro by doing it in a tube, and we have to kind of like mimic what happens naturally in the bacterial cell. Yeah, so we have cut smart buffer, which we used last time. We have ATP, we have the ligase or the enzyme, and then we have our DNA components. So the first DNA component is the intact one. And so the first thing we have to do is like we have to know, we know the target or the C2, the final concentration that we need for that. In our case, it's 50 nanograms. And so we just go back to our tube and get any of the intact plasmids. So far with our PCRs, we've been using, I think, the intact plasmid 1 or the mini prep 1. If I'm not sure if there's enough volume left right now, but if there isn't, you can always use 2 or 3. So just like we did with our purified PCR products, we have to check the concentration. I can't remember if we checked the concentration. Did we? I don't think we did. So anyway, we get that from the from -20 freezer, any of the intact plasmid. Let it thaw on the ice, and then just pipette like 1 or 2 microliters and check the concentration on the nanodrop, nanograms per microliter, right? And so, and then our goal is to get 50 nanograms. So like last time when I did it, the concentration for like colony 1 in the protocol was 66.5, and so using C1V1 equal to C2V2 to make 50 nanograms, I just had to use 0.7 microliter on that. So whatever it is, let's see how much volume we need to get out to get 50 nanograms. And then for the purified products, we have the concentrations, the volumes that we need for that. So those, yeah. So, and remember that we're doing go and get cloning for each, like in your case, for each gene. So there's one for 2 guide RNAs for one gene, 2 guide RNAs for the other gene. Just a second. Oh, I'm having these like minor contractions. They've definitely improved. Like they're not as painful. They're just uncomfortable. Okay. Okay. Yeah, so we get that, and then we put everything in one tube. So remember that the same thing we do about PCR, where you put all the reagents together. Always start with a bigger volume, which is always like water. So start with water. How much water do we need with all these different volumes, with the difference of 20? In our case here, like let's just say the water was like 12.9 microliter. Then you have to put 1 microliter of T4 ligase, 2 microliter of ATP, 2 microliter of Caspamide buffer, and then restriction enzyme is usually last, and then your purified product. So whatever volume you need, the biggest one first, and then the last one. Yeah, so we do that, and then just like a PCR, so we have all the different reagents, and then we add the volumes. You do one reaction for one gene, you put all these different things, one reaction for the different gene, different things. Once you're done, you flick to mix and also spin them down. And remember that for the restriction enzymes, before you add them into the tube, you should pipette them up and down. Just these two, you need to pipette up and down before, several times before adding, because you have to make sure it's really mixed. So once you have that, then you can put in the PCR machine, just like we do for PCR. But in this case, there are different conditions. And there'll be like much less, like fewer things than you need for a PCR. So the first stage has to be at 37 degrees. So you click on the degrees where it says like 95 in the PCR. So yeah, create a new protocol, press 37, and then press 5 minutes. So 5 minutes will just be 5.00. And then the next stage, 22 degrees for 5 minutes. And then next is 60 degrees for 5 minutes. The cycles, change them to 30. And then infinite hold, I think the default is 12. Change it to 4 degrees. And then you can save the protocol. But I think there might already be a protocol in there. Yeah, you can search for a protocol. It might be called Golden Gate Jetty or just Golden Gate in there. Okay, so that's for the Golden Gate reaction. So I'm excited. This is great because after this, that means we've created our own construct. We created our own construct for CRISPR, which is like, excuse me, the first big stage of this. The next stage after we create this, we then have to then transfer it into E. coli. So E. coli can now make many copies. So we're trying to transform E. coli with this new or recombinant DNA. So because it's a different combination of DNA pieces, it's going to help us like make many copies, then we put that into agrobacteria, and then we put that into medicago truncatula so that it can now do the CRISPR deletion. So yay, awesome. Let me know if you have any questions.